Hello everyone, I am honored to introduce the update of iNAP 2.0. It is a workflow that combines microbial metabolic interaction analysis with microbial network analysis. With the widespread adoption of metagenomic sequencing, new perspectives have emerged for studying microbial ecological networks, yielding metabolic evidence of interspecies interactions that traditional co-occurrence networks cannot infer. This protocol introduces the Integrated Network Analysis Pipeline 2.0, iNAP 2.0, which features an innovative metabolic complementarity network for microbial studies from metagenomic sequencing data. The main highlights of this pipeline are: 1. It provides a variety of methods for calculating microbial metabolic interactions. 2. The use of the RMT method provides an objective and reliable way to filter metabolic intensity. 3. It can identify potentially transferable metabolites between microbes, making it possible to establish a microbial metabolite bipartite network. The first section of the pipeline is to build a genome-scale metabolic model. This part provides 3 tools: using PROCA to predict protein sequences in the genome, using CARVM to automatically build a genome-scale metabolic model, and CARVM with gap filling. The second section of the pipeline is to infer pairwise interactions between GSMMs. iNAP 2.0 provides 3 methods to quantify metabolic interactions between GSMMs, PhyloMint, SMETANA, and FBA-based metabolic distances. Similar to the co-occurrence network based on species abundance, these metabolic interaction indices can be filtered and constructed into microbial metabolic networks. Among them, the potentially transferable metabolites between models predicted by the PhyloMint method are very important. The third section of the pipeline is to construct a metabolic interaction network. iNAP 2.0 provides a variety of methods to determine network thresholds, including methods based on random matrix theory, RMT. In the RMT method, in addition to the chi-square test method already in iNAP, a new RMT method based on the KS test has been added. In addition, for some values that are not suitable for the RMT method, we provide a method based on the z-score outlier detection to determine the network threshold. The fourth section of the pipeline is to analyze the metabolic interaction network. All the original iNAP tools introduced can be used to analyze the metabolic interaction network, such as global network properties, node centrality, modularity, and identification of module hub nodes. In addition, iNAP 2.0 also adds some new analysis tools for metabolic interaction networks. For example, find the intersection subnetwork of 2 networks, construct a bipartite network of microbe and potentially transferable metabolites, and plot a metabolic distance matrix heat map. As a case study, we apply this approach to a temperature gradient hot spring, unraveling the interplay between thermal stress and metabolic cooperation. Our analysis reveals a striking increase in the frequency of metabolic interactions with rising temperatures. Amino acids, coenzyme derivatives, and carbohydrates emerge as key exchange metabolites, forming the foundation for syntrophic dependencies, in which commensalistic interactions take a greater proportion than mutualistic ones. The mentioned study has been published on Nature Communications. Finally, let's make a summary. Here, we introduced the updated iNAP 2.0, whose main function is to integrate metabolic interaction analysis based on metagenomic sequencing data into molecular ecological networks. The updated iNAP 2.0 has added 20 new tools, allowing users to build and analyze metabolic interaction networks. The platform is open for registration, allowing users to upload data for de novo analysis or start analysis using intermediate data, and the platform provides corresponding demo data for researchers to learn and use.